Hello, dear students. I welcome you all on behalf of Sri Jayendra Rangjong classes. Dear student, we are in a continuation of the chapter that is plant breeding. And in plant breeding, we are at the end of this chapter and we are going to cover a last topic of this chapter, that is tissue culture in detail. Okay. So, tissue culture, the name itself suggests we are going to culture a tissue, we are going to form a tissue from a single cell. So, if we define it, then but what is tissue culture and what is the need of this? So as a traditional breeding techniques which we have studied till now in this chapter are inadequate, or we can say they are not sufficient to keep the pace with the demand to provide sufficiently fast and efficient system of crop improvement. Okay, so it is not able to complete the demand and fast and efficient system of crop improvement. Traditional breeding techniques have failed. So therefore, there is a need of another technology, and this another technology, that is tissue culture, was developed. So see how the tissue culture is done. Tissue culture is done in the like test tubes or a conical flask or like this in the laboratory. Now we have to understand what is tissue culture, what are the conditions required, and what are the different types of tissue culture that we need to discuss in this topic. So it was learned by scientists during 1950s that whole plant could be regenerated from the explant. Some scientists thought that in early 1950s that we can frame or can create a whole new plant from the a segment of a plant part that is called an explant. Okay. So the plant part which is going to be cultured, that is called as explant. Okay. So the explant is any part of the plant taken out and grown in a tissue under sterile condition. Sterile condition means micro-free environment. If any plant part taken out and grown in a tissue under sterile condition in a special nutrient medium for the production of new plant, then that is known as a explant. So it is a plant part which is going to produce a new plant in the aseptic condition, in the sterile condition, in the particularly a tissue culture technique. So see, this is the carrot. Suppose we have taken a segment of carrot, that is explant. We kept on the nutrient medium. This is a this is a petri dish. In this is the nutrient medium. In nutrient medium, we kept this explant. This explant grown or divided and formed a callus. We will discuss what is callus later on. Callus will form embryoid, then embryo will form, and embryo will form a new plant. Such a way, a tissue culture can be done. And here also you can see this is the excised piece of a leaf we can put, or direct we can isolate a cell and we can put on the petri dish, and this petri dish will form a new group of cells. So new plant production such a way can be carried out. So here the question is, what is tissue culture then? So if we define a plant tissue culture, then plant tissue culture, if we talk, this plant tissue culture, if we define, then it is PTC we can call, and it is known that plant tissue culture is maintenance and growth of plant cells, tissues and organ on a suitable culture medium in vitro. Now in vitro means in a container, in a artificial medium, that is test tube. Okay, so if any plant part, that is can be plant cell or tissue or organ, if that is maintained and grown on a culture medium in artificial condition, then that artificial technique is known as this plant tissue culture. And in that we use a explant, and we have studied now that explant is a plant part which is has a capacity to form a new body or a new plant. So here, if we talk about that ability of a living cell of a plant to give rise to the new plant, if one cell of the plant has a capacity to generate whole new plant, or explant has a capacity to generate whole new plant, then this capacity is known as totipotency. You can see here it is a living cell, but just it is divided and number of cells are formed, and later on it formed a new plant. So this capability of one cell to give rise to the whole new plant, that is known as totipotency, and that type of cells will be known as totipotent cells. Now here, to carry out this tissue culture, there is a certain environmental conditions are required. So now the important thing is environmental conditions required. If we talk about environmental conditions required, then there are three important aspects in vitro culture. There are three important aspects of in vitro culture, namely, number one, or the conditions required. Number one is nutrient medium. Number two is sterilization. These three things are required. Okay, and third, that is the aeration of the tissue. These three conditions, or we can say aspects of in vitro culture, are necessary to carry out the tissue culture. So we are going to discuss the three things: which type of nutrient medium is required, what is the sterilization, and how aeration is maintained. That we are going to discuss now. So nutrient medium, if we first talk about this nutrient medium, then it is known that it will provide nutrition that is required for the desired development of the explant. So it is going to provide a nutrition for the development of the explant. And this nutrient medium consists mainly of sucrose, which is going to act as a carbon source. Then some inorganic salts will be there, vitamins are there, amino acids are there, and growth regulators or growth promoters are also there, like oxygen and cytokinin. So all these things are required in a nutrient medium which is going to provide a nutrition to the explant. And the commonly used the growth regulators or growth hormone are auxin and cytokinin. Okay, in the particularly we can say this tissue culture medium. And in auxin also, if we talk 2,4-D is used. See, these are the chemicals which are going to be used. So now here the auxin which is used here, that is the 2,4-D, that is 2,4-dichlorophenoxyacetic acid, and cytokinin which is used here, that is a benzyl aminopurine, VAP. This type of auxin and cytokinin are added in the nutrient medium for providing the growth. And these growth regulators are required for the cell division and organ regeneration from the cultures. Now, culture medium, if we talk, that is rich in nutrient and therefore attract the growth of microorganism, thereby resulting in the contamination and the spoiling of the medium. So sometimes it also happens that the particular we are going to grow a particular desired bacteria or desired organism or desired tissue on the petri plates, but some microbes contaminate it. So our target or desired plant is not going to be generated. Our desired explant cannot be grow properly. So that is the important thing. So for this, the sterilization technique are required. But we remember the culture medium may be liquid or solid. This nutrient medium, if we talk, that may be solid or liquid. It can be solid or liquid. So apart from this, now the culture medium it need to be sterilized to remove the unwanted microbe and to retain it from the contamination. So second thing which is required, that is sterilization. So therefore culture medium is sterilized to kill microorganism, and sterilization of nutrient medium is done in an autoclave, a steam sterilization. We remember it is done in the steam sterilizer. So this is the autoclave machine. In this is the opening part. We remove this open, and we put a test tubes and conical flask and culture medium, all this thing in this, and we particularly heat it. So and we fill a water level up to this, and overstand this, all things are put inside. So what will happen, the steam will be generated, and under high pressure and at a high temperature, under a steam, the particular microbes are going to be killed by this particular sterilization. So killing of microbes associated with the nutrient medium is called as sterilization. So here ultimately sterilization is need to be create a particular thing, microbe free environment. Okay. Now the culture medium is autoclaved for a 15 minute at 121 degrees Celsius and 15 pounds of pressure. Okay. So that is in the inside this autoclave. 121 degrees Celsius temperature is going to be maintained, and 15 psi pressure is going to be maintained, and this is carried out for 15 to 20 minutes. So this way sterilization can be done. So if we talk about sterilization, it is known that sterilization is is essential that the the explant that the explant culture vessel, media, and instruments used for tissue culture be made free from microbes. So to make a free from microbe explant culture vessels, media and instrument, the sterilization is required. And for this purpose, two types of sterilization is done. It is of two type. Number one, surface sterilization. And in surface sterilization, if we talk, explants is treated with specific antimicrobial chemical. Suppose I am taking one conical flask and I have taken a stem explant. Since a pieces of stem which is going to be taken an explant is added here, and along with I am adding 0.1% HgCl2, which is an antimicrobial agent. So that HgCl2 will particularly make a surface of this explant free from the microbe. So this is our surface sterilization which is done with the explant. Now after this surface sterilization, another type of sterilization which is carried out, that is known as complete sterilization. And in complete sterilization, if we talk, then it is known that the vessels media and instrument are suitably treated with steam under autoclave. Okay, so vessels, media, and instrument are suitably treated with a steam autoclave, then dry heat or alcohol, or subjected to filtration to make them free from microbe. And this is called a complete sterilization. This is called a complete sterilization. And apart from this, the third thing which is required, that is proper aeration of cultured tissue is also important, is also important aspect of tissue culture technique. So why proper aeration is required? Because when proper aeration will given to the particular microbe, or we can say the particular explant, sorry, not microbe, explant, then this explant will get a proper aeration. It will respire, some cells will respire, and it will get energy, and so its division will be proper. Now after this sterilization, we are going to talk about the methods of plant tissue culture. And when we talk about the methods of plant tissue culture, in the methods of plant tissue culture, we are going to talk about first, that is, it has a two methods. Okay, so two methods. One is type of in vitro method. So in this first we are going to talk about the methods and the type of in vitro growth methods. Means the plant tissue culture, in this there are two methods. One is based on the type of in vitro growth, how they are grown, and in this there are two types. In this we study about the callus culture and suspension culture. And the B type, a second method in which we are going to talk about the types of tissue culture based on the explant, a type of the explant. Okay, so in this the second method is based on type of explant taken. And if we study based on the type of explant taken, here we will study in this about the meristem culture, protoplast culture, embryo culture, etc. We will study based on the type of explant involved. So first we are going to talk about the type of in vitro method, and in this we are going to discuss a callus culture and suspension culture. So when we transfer a segment of DNA, a segment of explant on the sterilized nutrient medium is called inoculation. When first time we pick a small segment of plant part, that is explant, and put it on the culture medium, it is known as inoculation. So it is done in an aseptic environment, that is in the micro-free environment, that is in the laminar airflow. And inoculation chamber or laminar airflow that look like this. See, this is the particular chamber you can see here. These are the laminar airflow. Okay, and in this laminar airflow, UV light is inserted, and along with this, the particularly we can say the alcohol, we wash the platform with the ethyl 70% ethyl alcohol, and before starting the experiment we on this UV light for the something 20 to 30 minutes, so all the microbes in this particular region get killed. Okay, like this laminar airflow is sterilized first, and in this sterilized medium the inoculation is done and all the tissue culture technique is carried out. See, this is the proper way it is looking. See, up to this is all side it is fitted with the glass, and here it is, this glass slide is up to this layer, only this part is open. Okay, and we open this only it is slider part, so this slider, see you can see this slider is upward. Before initiating the experiment, when you view on UV light, we close this slider and we make a proper sterilization. It allow to carry out a proper sterilization. Okay, so this is done. Now in callus culture, if we talk what is done, so first we are going to talk about the callus culture. And in callus cultures, what do? We take a particular conical flask or a petri dish, and on this we provide a proper nutrient. It is a nutrient medium, and on nutrient medium we inoculate a explant. Now this explant is a small piece of plant part. Now this small piece of plant part in a nutrient medium. This nutrient medium has agar, which is act as a solidifying agent. So this nutrient medium will be solidified, and on this the explant is kept. Now the explant will divide, and after division it will form a mass of cell, and this mass of cell is known as callus. So this way the callus is developed on this solid medium. Okay, so this callus is, it is unorganized, undifferentiated, it is unorganized undifferentiated mass of cell, and this unorganized undifferentiated mass of cell that is formed from the explant by division of it, and it is maintained on a medium gel with agar, so it is a solid medium, and this medium also contain the 2,4-D and benzylaminopurin as a auxin and cytokinin. Now, with the passage of time in this, when the callus is formed, a cell or tissue dry up. This cell or tissue dry matter increases, and the level of nutrient decreases. Okay, okay. So this will happen in this callus culture. So this is occur in the callus culture. Okay. So how this nutrient maintenance is maintained, that we will discuss in the class. This is the callus culture. So the culture are incubated for 3 to 4 weeks. During this period, the cells of explant absorb nutrient, grow and undergo repeated divisions, and this produce a proliferated undifferentiated mass of cell that is known as callus. So callus is formed from the particular by absorbing a nutrient at 3 to 4 weeks. It will take time. You can see this is the, we have inoculated with the small piece of mass, but now it is a this much. So this is a callus. Okay. Now after this, sometimes shoot or root may be produced directly without the formation of calyx. It also takes place that a shoot and a root directly form without the formation of calyx. So the explant or the callus cultured on the different combination of auxin and cytokinin to produce a shoot and root. So different combination of auxin and cytokinin are used in the nutrient medium, which will allow to produce a shoot and root. And we know that if the auxin concentration more, root initiation will occur, and if the cytokinin concentration is more, shoot initiation will occur. So the production of shoot and root from the callus is called organogenesis. Okay, so shoot and root formation is known as organogenesis. Okay, and in organogenesis, you can see such type of organs will develop. See, initially it is a callus, but later on this organ particularly will develop, and shoot and root will be formed. So this is a shoot, and this is a root. This is organogenesis. Okay, now this is callus culture. Now second type we have discussed. We discuss here. The second type is suspension culture. And in suspension culture, if we talk here, the explant is suspended in liquid medium containing 2,4-D, that is a type of oxygen. Okay, so what here we do, we take a conical flask, and in conical flask a liquid medium is taken, and on this liquid medium the explant is kept inside it. Now this explant gets suspended in this liquid medium, and later on this is going to form initially root part, and some initial explant will grow and form this way the culturing, and the new plant part will be developed. So such a way, when the suspension in liquid medium is done and the culturing happens or new plant part develops, then it is known as suspension culturing. Now this culture having explant, having explant is constantly agitated, is constantly agitated at a speed of 100 to 250 rpm, and this rpm are revolutions per minute. So at 100 to 250 rpm revolutions per minute, this constantly agitated because so proper, we can say the nutrient they can gain in proper, they cannot form a clump. So clumping, to avoid a clumping, this revolution or agitation is done. Now when we talk about this both type of tissue culture, it is known that in both the type of tissue culture, that is callus culture and suspension culture, if we see in this cell or tissue, dry matter increases. That cell or tissue dry matter get increased, and level of nutrient in culture medium get decreased. So dry matter or a mass or callus or organ formation is developing, so it is increasing, and along with the passage of time, the nutrient are going to be used and absorbed, so the nutrient are getting decreased. So if at, so after some time the time will come when there is a no nutrient. So if nutrient will be diminished or decreased, it will damage the newly formed cells. So to prevent damage of newly formed cells, due to decrease in the level of nutrient, subculturing is done. So now the question is, what is subculturing? So if we talk about a subculturing, what is done in this? Let us discuss. Now in subculturing, what we will do, suppose this is a petri dish, this is a nutrient medium, and on this is a callus. Now callus is growing, so nutrient in this are getting decreased. So time will come that nutrition are completely over, and it will start to use the own cells, own callus cells. So what we will do? We will take a fresh new petri dish. So fresh or new culture plate we will take, and this new culture plate have the nutrient medium which is similar to this. So nutrient medium in both the plates are same. Now we cut a piece of segment from here and put here. So now it will start developing and it will form a new callus. So the damage of tissue can be prevented. And here also nutrients are getting used up. So this way, with the passage of time, to prevent the damage of newly formed cells in subculturing, if we talk, so like this subculturing takes place, we take a piece of the newly from the what a particularly damaging plant part or in which the nutrient medium is over and put in the newly culture plate having the same nutrient medium and allow it to grow. Such a way subculturing is done to prevent the damage of newly formed cells. So if we want to define it, what is subculturing? So in this part of culture, in this a part of culture are regularly transferred to a new culture vessel, transferred to new culture plate or a vessel containing fresh media. Okay, if you carry out this, then it is known as a subculture. Okay, so it is a subculturing which occur like this to prevent damage of the media. Now in this, you will see that the shoot and root regeneration will occur, and it is known that shoot and root regeneration are are generally controlled by auxin cytokinin balance. And by auxin cytokinin balance, the shoot and root regeneration will occur. And we know that if there is a excess of auxin in culture media, it promotes root regeneration, and excess of cytokinin promote shoot regeneration. Okay, so root and shoot regeneration occur by based on the which one is more in the culture medium. Now in this, how the culturing done? So first, callus culture are first kept in benzyl aminopurine containing medium. So what we do? We will take a petri dish, and in this a callus is kept, and this has excess of benzyl aminopurine, that is a cytokinin. So what it will do? They should regenerate from the calyx. Should regenerate. So should regenerate. Okay, from the callus. Then should regenerate from the callus when the should become 2 to 3 centimeter long. So should regenerate from the callus. Okay. Now when shoot become 2 to 3 centimeter long, they are excised or removed from culture plate and transferred to oxygen containing medium. Now in this oxygen containing medium, root will regenerate. So next point is, in oxygen containing medium, root regenerate from lower ends of this shoot to yield complete plantlet. So it is going to be look like, suppose we are doing it in a conical flask or in this particular petri dish, then it will going to develop such way plantlet and root. Okay. Now after the development of complete plantlet, this whole plantlet is removed from this oxygen containing culture plate and established in field. In field. And this established on field is known as hardening. So hardening is done like this. And what is hardening? The transfer of, it is a transfer of plantlet from culture vessel to field or soil. Now we remember that this hardening, during hardening or during this process, plantlet are kept under reduced light, and high humidity. So the growth can be proper. And due to the hardening, this hardening processor make the plantlet capable of tolerating the relatively harsh environment outside the culture vessel. And slowly, this plantlet will develop as a new plant in the soil. Okay, so this is the what a tissue culture method, that is a callus culture and suspension culture, which are done in the artificial condition in the lab. Now we are going to talk about after this about the type based on the, particularly we can say, the explant. And so the part 2 of this we will continue in the next lecture. We will study the type of tissue culture based on the type of explant. Till that, thank you everyone.